Okay, great. Well, thank you everybody again for joining us for the International COVID-19 HPC Knowledge Exchange, our now monthly webinar series that allows us to people from all over the world to be able to present some results, in progress results, wherever you're at, about and what the work you're doing to spur collaborations and cross-talk about COVID-19 research. My name is Sean Brown. I'm the director of the Pittsburgh Supercomputing Center, representing Exceed in this forum. And thank you all for joining us. I'll give my counterpart at Praise the chance to introduce himself.
Yeah, hello, I'm Matej Prapnik. I'm a professor of physics at the University of Ljubljana in Slovenia, and also head of theory department at the National Institute of Chemistry, also in Ljubljana. And I am the chair of Praise Scientific Steering Committee, which Praise is the counterpart of Exceed in Europe. So I also welcome you to this webinar series. And today we have two talks. Sean, I think you will introduce the first one.
Yeah, I just want to say the PI of Exceed is here with us, John Towns, for the first time in a while. I know he's always busy, so it's great that you joined us. Thanks for joining. We have two really good presentations today. I'm really excited to hear about the progress that's going on in the COVID-19 world. So our first presentation will be given by Victor Padilla-Sanchez, research scientist at the Catholic University of America, and Theresa Deisher, who's the president and CEO of ABM Biotechnology. So I'll let you guys take it away. You should be able to share your screen, Victor.
Okay. One moment, please. Share screen. Share. Yes. Can you see it?
It's either coming or right now I'm seeing a black screen.
Oh, wow. It says it's share.
Do you have a monitor as well? Sometimes you have to disconnect an outside monitor on Zoom.
No, it's a laptop.
It's just a laptop?
Yes.
Looks to me like a specific application was shared instead of the screen.
Yeah, looked like other windows were on it.
Oh, that's funny. Sorry for that, but
It's okay.
Alternatively, what I can do is join from my other computer and put the slides there. So, and then talk by my laptop.
Okay.
Yeah, hopefully that will work. Share screen. Okay. Can you see that's the PowerPoint?
No, still can't see anything. Maybe stop sharing and reshare.
Okay.
Yeah.
Oh, someone. Okay, wait. So, someone is sharing here. So stop share here. And now?
Now you've turned off sharing.
Now we see you. Okay.
Where is that? Here. Share.
It's coming.
Yeah.
No, still seeing a black screen, Victor. Sorry.
Oh, I'm so sorry, but yeah. Can you go ahead and start, Teresa?
Yeah, I can while you try to share your screen, and I'll just give people a background. So ABM Biotechnology is working on a small molecule drug that, as its mechanism of action, mobilizes what we call supercharged cells into the bloodstream. And we call these cells supercharged because the natural killer T cell and the cytotoxic T cell that we mobilize express CD3 at a log higher than classical T cells or classical natural killer T cells. And CD3 determines how, you know, activated and effective these cells can be. So natural killer T cells, cytotoxic T cells, you know, by nature like to target and kill abnormal cells, whether it's a cancer cell or it's a virally infected cell. And we are going into clinic both for relapsed refractory non-Hodgkin's lymphoma, as well as for COVID or influenza-induced moderate to severe acute respiratory distress syndrome. And with the biology that has really been learned about the COVID-19 patients, where monocytes and lymphocytes are chronically infected, those activated infected monocytes and neutrophils are driving the lung pathology. And we know that immune imbalance also triggers coagulopathy as well as you know, multi-organ failure. And this is sort of the composite of what's being seen in these patients. We became interested in the receptor target of our drug as a potential receptor for COVID-19. It's also expressed on bronchioles, and the Human Proteome Project has validated the expression of this receptor, which is ICAM3, and the data from our labs. So ICAM3 is very selectively expressed on monocytes, lymphocytes, less densely on neutrophils, on cancer cells, and then on human bronchioles. And because of the biology that's really targeting these specific cells, we wanted to know if it could be a potential secondary receptor for ICAM3. It's really important that we identify all of the receptors in HIV. You know, back in the 1980s, there were blinders and a focus on CD4. And because there were additional receptors, you know, vaccines, antibody candidates, and a lot of the early therapeutics failed because they were not addressing all of the means for entry into cells. So we did some preliminary molecular docking and saw low energy binding of the SARS-CoV-2 spike to the ICAM3 IG1 domain. And then we reached out to this group to actually look for people with expertise in molecular modeling who could do some, you know, more expert modeling and either confirm or refute what we saw. So Victor has done quite a bit of modeling and has some really interesting results. And he'll show you, but it does look like ICAM3 has a high affinity binding for SARS-CoV-2 that's equivalent to the human ACE2. And then I don't have data today, but we also have some thermodynamic data, and the KD for binding is about 10 nanomolar. That's from Frank Zhang's group. And similar to the KD binding to the human ACE2. So we really would like to reach out and try to see in the group if there are people with in vitro assays or in vivo assays that could help us with some follow-on experiments to really look and see the binding to ICAM3 versus the human ACE2. So Sean looks like he has shared the slides, and I think those are probably the slides that Victor will go over.
Yes, I can see in the screen the slides.
Okay, great. Yeah. Just let me know when you want me to switch slide.
So can we go to the first slide? Yes. So we were asking for in vivo or in vitro assays, and that data in the second slide.
That one?
If you can switch to the second slide.
This is the second slide.
No, this is the
Yeah,
first.
still at the first slide, Sean.
Oh, let me, are you seeing, I know you have the swap displays, hang on. Is that better?
I think you have to click on the next slide. This first slide is still up. We can see in the window on the left the additional slides.
Okay, it's, yeah, so, okay. Oh, weird. Swap percenter.
There you go.
Okay, I'll just do that.
Yes.
Okay, there, you can see it.
So, sorry for that. So the docking, the binding with Rosetta. So what I use is Stampede 2 from Texas, which is a supercomputer, which is very fast. And it shows a binding between RBD and ACE2 at minus 13.607. So that's the interface score. And that's between ACE2 and RBD, which RBD is in blue for SARS-CoV-2, and ACE2 is magenta. So that's the main thing. And then for the next slide, yes. So what I have done is first use class pro to find a probable location of binding between ICAM3 D1, domain 1, and RBD. And I found that the probable location of binding, and then I input to Rosetta. So for Rosetta, I found an interface score of minus 15.024, which is better than ACE2, which is very good. Now you can see on the right of the slide on top the graph of the models that are constructed, and we find that's the benchmark of a good docking. So they point to the left, to the bottom. Then I analyzed some interactions, and I found that hydrophobic interactions are clustered in the middle of the interface, while we have salt breaches on the sides. So that should point to that the interactions are strong. I use two servers, PDB, PISA, which is very well established for hydrogen bonds and salt breaches. And then another server to find more interactions like hydrophobic interactions, called check. Plus, so the main point is, according to these results, ICAM3 binds better than ACE2. And the next slide, can you point the next slide?
Yep, there you go.
Okay, so the next slide is SARS-CoV. The previous was SARS-CoV-2, this is SARS-CoV. Again, I did class pro for the best location of binding for input to Rosetta. Yes, and then I ran Rosetta doc. We have one salt bridge, few salt bridges, less hydrogen bonds, less hydrophobic interactions for an interface score of minus 9.602. So very much less than the previous two for ICAM3 or ACE2. So that's what we have. Of course, this is a very simplified version, a very simplified explanation of what I have done. It takes a long time. So, how many minutes do I have right now?
Oh, you're fine so far.
Sorry?
So Victor, I just want to add the purpose of looking at the SARS binding is that the human ACE actually binds well to both the SARS-CoV as well as the SARS-CoV-2 spike. But the ICAM3, binding really is reserved from these models to the SARS-CoV-2 spike. With this data, we're really just trying to reach out to find people who have established models, to verify this with biology. And I think, that's really important for people to have this information and to know about this interaction. There are some concerns about the vaccine approaches to the spike, and on our board are two vaccine experts, Manon Cox and Gary Groman. And one concern about vaccines to the spike is that the antibodies generated could actually interact with natural ligands to ICAM3 and lead to, chronic alterations in the immune system. So it's interesting, in those severely ill COVID-19 patients, they develop antibodies within 7 days, and yet they go on to severe disease. So the development of those antibodies is not providing them protection. And based on some of these novel, interactions, we're concerned that the antibodies themselves could be driving the imbalanced immune system. And so particularly where efforts are going to be focused in vaccine development, I think it's really important that we understand how this ICAM3 interaction is going to play in to the biology.
Great. Does anybody have any comments or questions you'd like to ask Victor and Theresa? Or some insight into that, to that question that Theresa just posed?
Hi, this is Eve Worley. I don't have a video because our power was out. Theresa, and Victor, your work is so interesting. Like, it's just like, wow. And I'm not a human biologist, but so the ICAM3, like, there's a lot of people with antibodies that are doing quite well. Are you testing, like, or is somebody involved in testing to see what these different antibodies are interacting with? Like, are the antibodies interacting to different things other than the parts of spike that might bind to ICAM3, or is there somebody doing that?
They're not yet, but obviously we would like to see if the antibodies to the spike interact with the DC or L sign, which are the natural ligands for ICAM3 and are found on dendritic cells and other immune cells. But I should reach out to the antibody people. It's a good idea.
So I have one more not follow-up question, but a different question. So what our work is focused on is looking at orphan genes or novel genes that might be either human specific or even ancestry specific, and we're identifying a number of transcripts that are expressed differentially between, for example, African American and European American populations that are specific to COVID infections. And are you looking at anything like that? Is that any part of what you're doing?
No, you know, we're not. We're a small company and we're focused on the clinical development. But the, you know, African Americans are getting much more severe disease, and that may be because of, you know, differences in gene expression. There are differences in their monocytes. So we are hoping that our therapy is going to have a profound effect to help those people who are more susceptible and really getting severe disease.
Okay. Well, I love your research, and after hearing your talk, I think we'll look specifically at some of the genes that you're describing, and especially ICAM3. And if we have anything that pops up that's interesting, we'll let you know right away. So.
Okay, thank you. That would be great.
Okay, thanks. Thanks, Eve. Any other comments or questions? You can either post them in the chat or you can unmute and ask directly if you like.
I would add there is an ICAM3 mutation. I'd have to pull the paper. I don't know the exact site. That in one population, it actually makes them less susceptible to acute respiratory distress syndrome. So that ICAM3, you know, does seem to play some sort of role in ARDS. So it would be interesting if you find something, and we'd love to hear about it.
Yeah, I remember last night I was reading. I'll post it to the Slack when I find it. There was a study that was done where they did a large cohort of. It was in collaboration with China, where they did a large cohort study of the severely ill people that with COVID, and they found, I believe it was 3 proteins that were common in all the severely ill people. I just read it last night, so I'll post that to the Slack. There may be some insight there too. Not exactly the same thing that you're looking at, but it might have some consequence that I just can't remember. It was on my news feed, so I'd have to go back and flip through Flipboard like 700 times to find it. I didn't save it.
Great, that would be great. Mm-hmm. All right, well, thank you. I'm going to have to jump off onto another meeting. And Robert, I'll respond to you a little bit later.
Okay, great. Well, thank you very much, Theresa and Victor. Really appreciate the presentation, and look forward to more findings as they come. And if anybody wants to contact them, remember we have the Slack channel, and you can directly email them as well, I'm sure. So thank you again for the presentation. Really appreciate it.
Thank you.
Thanks.
And I will stop my screen share. And so our next presentation is from Giulia Palermo, who is, I have to go get my notes. I'm sorry. I don't have it memorized. I'm sorry.
Professor of Bioengineering.
Yes, assistant professor of bioengineering at University of California, Riverside. So thank you, Giulia, for coming to talk to us, and we look forward to the presentation.
Thank you very much for this opportunity to present here some of the work that we are doing to aid the detection of SARS-CoV-2. So I should mention that these are really preliminary results obtained through classical molecular dynamics simulations, which actually have posed the hypothesis together with literature data for the simulations that we are performing on Anton, which I will also update you. So we are studying the mechanism by which this system called CRISPR-Cas12a, which is a novel CRISPR system, is used to aid the detection of viral nucleic acids, and in particular emerged this year very recently to be useful for the SARS-CoV-2 detection. So I think that many of you have heard about the revolution occurring in 2012 when it has been discovered that a protein binding to nucleic acids was able to efficiently perform genome editing in a programmable way. So the way how this CRISPR system work is that they have developed an immune system, bacteria have developed an immune system to defend from viral infection. So what happens is that there are a series of proteins. The most famous is Cas9, which we have studied extensively, binding to a guide RNA which arises from the processing of viral nucleic acids. And in this way, the protein has basically remembers the viral infection and can target, thanks to this guide RNA, the viral nucleic acids for deactivation, for double-stranded cleavages, and there be deactivation of the viral infection. So this system, CRISPR-Cas9, has been used for genome editing in several applications from biology, biotechnology, medicines, and even material science. But now, very recently, there is a new CRISPR system called Cas12a, which appeared to be very useful for viral detection. So what happens is that the viral RNA from SARS-CoV-2 is basically amplified and through PCR, and the amplified synthesized viral DNA binds Cas12a and give rise to a first cleavage. So this is called cis cleavage, but the detection actually occurs upon a second biophysical event, which is called transcleavage. The transcleavage is basically the cleavage of single-stranded DNA that can access the active site upon opening, upon a conformational change. And since these single-stranded DNA are attached to a fluorophore, they can basically allow the signaling. So by using the COVID-19 HPC consortium resources, in particular COMET and Anton, we are studying these conformational changes, and I would say to get insights for both cis cleavage, so for both what should happen to allow the binding and cleavage of these synthesized viral DNA, and also, most importantly, for the conformational change underlying transcleavage. Something else I should mention is that this conformational change for transcleavage, which is a bottleneck for the SARS-CoV-2 detection, and this has been actually proven through single molecule experiments to be the rate limiting step of the entire process, and also to be sufficiently fast to be detected through multi-microsecond length molecular dynamics simulations. So in this slide, this is just to show you why this methodology, the CRISPR-Cas SARS-CoV-2 detection, is becoming so popular. Is because it is easy to implement, is very rapid, and is accurate, and actually recently received also the FDA approval. This is a slide to show you that we have previously employed Anton to study the conformational activation of CRISPR-Cas9, which is the first CRISPR tool found to be active for genome editing. So we used Anton basically revealing a large conformational change of this domain HNH docking at the cleavage site to enable the genome editing process. So we started from a crystallographic structure. The system rapidly folded into a docked state where HNH is at the level of the DNA strand for cleavage. What also was happening is a large conformational change of this recognition lobe. What is important is that when we started these simulations, we started from this pre-activated state, we ended up into this activated state, which was in agreement with existing single molecule threat data. However, only a couple of years later, we had information of the fully active state from cryo-EM, actually finding that it was in actually superposable to what we found with Anton 2. And it was very nice because they also recognized the capability of these simulations to achieve an activated state. But let's see what should happen in Cas12a. So this is a movie showing the entire process of cleavage and of cis cleavage, and trans cleavage. Seems not working. Eccolo. So first you have the guide RNA binding, then you have the DNA which comes from the PCR that should basically bind Cas12, and then we have the cis cleavage. Then rapidly a conformational change should occur, allowing the entrance of the single-stranded DNA for trans cleavage. So this conformational change, which should in principle involve one portion of the protein called NUC, is what we are investigating with Anton. But to get initial result, to see if this study would have been feasible on Anton, we also performed classical molecular dynamics simulations in-house. So we started our investigations with a basic classical MD investigations, considering the system, the available crystal structures bound to RNA and bound to DNA from different species. So you see the system is composed of multiple domains, in particular a recognition lobe, which basically binds the guide RNA and allows also binding of the so-called target DNA strand. And then we have a nuclease lobe, which is located in this region. The cleavage occurs at the level of this active site in RABC. Then we have an additional domain, which is called NUC, which was initially thought to be responsible for the cleavage of the second strand of DNA, but then it was proven to biochemical experiments that actually NUC is not catalytic. So one of the main questions that was present in the literature, which we are also studying right now, is what is the role of NUC? If it is not catalytic, what is NUC doing in this system? And most importantly, the most important question is, okay, the cleavage of DNA occurs here at this level, at this active site. So it's intuitive to understand that the cleavage of the non-target strand would occur here, but it's not intuitive to understand how the cleavage of the target strand, which is in cyan, would occur in this active site, and how the cleavage of the trans cleavage of this single-stranded DNA would occur. So preliminary classical molecular dynamics simulations performed for the system bound to RNA and to DNA could give us already some insights on this process. First of all, we performed very traditional analysis, such as root mean square fluctuations, principal component analysis, and this could tell us, these are the fluctuations over time. You could see that in the RNA-bound form, the PAM interacting domain, so the domain that is responsible for the recognition of DNA, is very flexible. But upon binding of DNA, the motions of this portion are quenched, and actually the motions are transferred through REC2 and NUC, which is actually, I would say, pretty remarkable, because if you think that the binding of DNA, especially in this region, should actually quench the motions of the interacting domain, rather in this case, we have an increase in motions of domain. So the coming out message from these simulations was that after you bind DNA, we have a switch in flexibility from this region to this peripheral region. By performing principal component analysis on these trajectories, we could find that prior DNA binding, you could have this type of motions of the PAM interacting domain basically pointing in, but upon DNA binding, again, the motion was transferred to REC2 and NUC. This observing also an open to close conformational transition, which you can observe basically in this in this representation here, where you see the starting structure and what happens along the first principal component, along the essential dynamics. You have the open to close conformational transition. We also performed analysis of the correlated motions, in particular of the traditional cross-correlated motions, so Pearson coefficients, and the generalized correlations, which take into consideration Shannon entropy and are more descriptive of correlated motions that occur actually in different directions, not only, I would say, orthogonal with each other. So what we could observe is that upon DNA binding, in particular upon binding of the first strand and then the second strand, we have an activation of the motions, which is very interesting because in particular we have a turn on of the motions of REC2 and NUC, and upon binding of a complete non-target strand, we see that the entire system actually is turned on in motion, which actually could give us idea of an allosteric communication activated upon DNA binding, which we are also currently investigating. If we sum up these correlated motions into our classical, yearly classical MD simulations, we can clearly see that upon DNA binding, there is the activation in the motions of NUC and REC2, which is actually very remarkable, is observed in 4 independent replica of 1 microsecond each, and basically they confirm this observation of the single replica. This is particularly interesting, the motion, the correlated motion, the coupled motions of REC2 and NUC, because this was actually observed also in CRISPR-Cas9 prior we performed Anton 2 simulations on the system. So what you can observe is that HNH, which is the counterpart of NUC in CRISPR-Cas9, moves concertantly with REC2 and REC2 in CRISPR-Cas9. And upon these results, by using Anton, we basically could determine the full activation of HNH. In particular, in Cas9, what was important is that the motions of REC2 regulate the HNH in the activation process. And this was actually also found through single molecule FRET experiment, revealing that REC2 and HNH actually have a coupled dynamical function to ensure catalytic competence. So this suggests taking in consideration also what we observe in Cas12a, and considering also that I have to say for Cas12a, single molecule FRET experiments looking at these motions are still absent. So this suggests that in Cas12a, REC2 could regulate the activation of NUC in a similar manner as we observed in Cas9. Something that was also particularly interesting is when we look at the dynamics of the system before the non-target strand cleavage, which is this strand, and after the non-target strand cleavage. So I have to mention that biochemically, it has been proven that the system first cleaves the non-target strand, then has to cleave the target strand, likely to a conformational change of this area to basically bring together the catalytic site and the target strand. And finally, as a third event, as a third biophysical event, there should be the transcleavage of the incoming single-stranded DNA. So by looking at the dynamics of Cas12a before the non-target strand cleavage and after the non-target strand cleavage, we could observe an expansion of the binding, of the cleaving, of the cleavage site before the non-target strand cleavage, and a contraction of the active site after the non-target strand cleavage. So this basically could allow us to speculate that since Cas12a cleaves First, the non-target strand, a conformational change of NUC. And in this case, now that we have looked at the coupled dynamics of REC2 and NUC, so aided by REC2 would allow the approach of the target strand into the active site for cleavage. So now this has opened the hypothesis for performing Anton2 simulations, which are running to basically investigate how NUC could aid the cleavage of the target strand. So if I could mention some preliminary results which are not reported because the simulations are running, Anton is actually giving very good information for both cis cleavage and trans cleavage. So I have to say that in the case of trans cleavage, Anton is showing the opening of NUC with respect to the active site to allow the entrance of the single-stranded DNA, which we are also going to investigate. For what concerns instead cis cleavage, we are planning some other approaches. In particular, we are planning both, of course, I would say targeted MD, because it's one of the most straightforward approaches when you want to observe a large conformational change, but a large conformational change, which is not actually immediate to classical MD, but we are also planning to apply metadynamics, because we have observed in Anton, using Anton, that there is an alpha helix which folds and unfolds, and actually could be, in our opinion, the bottleneck of this mechanism, the key actor allowing this opening and closure. And with metadynamics, we would like to compute the free energy surface associated to the formation of this alpha helix and the losing of this alpha helix. So I actually would like to share these ideas to basically see your perspectives on it. So this was the conclusion, and with this I want to thank, first of all, my group that is working on these simulations. Akash Saha is a graduate student in my lab who is one of the people that found the importance of this transcleavage mechanism. Then we have Lukasz Niercwicki and Pablo Arantes, which are two postdocs in my lab. Pablo is performing the simulations on Anton2, and Lukasz is actually performing the enhanced sampling and metadynamics simulations. And then, of course, other group members and Martin Ginek, which is our experimental partner at the University of Zurich. So thank you very much. And of course, the computer time, which is actually essential for these simulations XIDE and the COVID-19 HPC consortium. Thank you very much.
Great, thanks, Giulia. That was great. That exciting work, very exciting work. Does anybody have any questions or comments that they'd like to make? You can either type in the chat window, or you can unmute and ask a question directly. No questions, no comments? I guess it was so clear that everybody got, but it was very exciting work. Yeah, it's always nice to see what Anton 2 what people can accomplish with Anton 2. It's an amazing machine for long scale molecular dynamics. Okay, well, people, I know, Giulia, I believe you joined the Slack channel, or we can send you the Slack channel link, but if anybody wants to contact Giulia by email or the Slack channel link, please feel free to do so. Keep the conversation up. Thanks again for a really nice presentation, and we'll hopefully, we'll be going back to, on September 10th, we'll be having our next webinar. Hopefully we'll have a lot more participation because Europe will be back from holiday, which will be nice. And I know we've got like 20 people that want to present there, so we may have to start gatekeeping.
I think then it will be just the opposite situation as now. Yeah.
Right. So, well, thank you all for joining us, and we'll see you next month. And as usual, keep the conversation up on Slack and on the email list, and we'll keep trying to fight the good battle. Thanks, everybody.
Thank you very much. Bye-bye.
Thank you very much. Bye.
Okay.